you, Vishal. A very good evening to you all. I welcome you in this amazing and informative talk organized by Delhi Student Unit, Microbiologists Society, India. I'm very thankful to Professor Arvind Deshmukh sir and Dr. Archana ma'am for always encouraging us to organize such valuable talks. I, Ankit, a final year student of BSc Honours Microbiology at Swami Shraddhanand College, going to convene this webinar with two of my worthy classmates, Anshita and Abhikash. Flow cytometry is a technology that provides rapid multi-parametric analysis of single cells in the solution. Flow cytometry utilizes laser as a light source to produce both scattered and fluorescent light signals that are read by detectors like photodiodes. A variety of fluorescent reagents are utilized in flow cytometry, include fluorescent conjugated antibodies, DNA binding dyes, etc. It is a powerful tool having application in immunology, molecular biology, cancer biology, etc. To share such more exciting information with us, our today's esteemed speaker is here, Mr. Pradeep Kumar, sir. He's a flow cytometry and equipment specialist. Sir is a super senior from our college, and we are proud to have such seniors like him. Sir is going to talk about what is flow cytometry, its basics, applications, and most important, the employment opportunities. So to start with the event, I would firstly like to invite our pillar of strength, Professor A.M. Deshmukh sir, the All India President of MSI. Please, sir, kindly inaugurate the session.
Thank you, Brahmaji and team, because Brahmaji is not alone. He is always supported by the team of microbiologists from the students of Delhi chapter. So, at the beginning, I would like to appreciate Dr. Archana ma'am. The reason is that every day, every week, every month, she is collecting diamonds in the basket of Delhi subunit, Delhi chapter of Microbiologists Society. And ultimately, it is a benefit to the central unit of Microbiologists Society. Today the topic is excellent, as Brahmajit has told. So flow cytometry, and the more important, which I like much, is job opportunities. Also, he is going to discuss. So that's a very nice, and step by step we are doing progress, and in the progress of Microbiology Society, the role of Delhi chapter is definitely the best role is played by Delhi chapter. So it's my duty as a president of Microbiology Society to be, I'm witnessing each and every lecture and appreciating the efforts by Delhi chapter. So as a president of Microbiology Society, I wish all the best to Dr. Pradeep and Dr. Archana also. For the listener, again I will request, listen carefully and percolate this knowledge up to others by grasping it today. Thank you very much.
Thank you so much, sir, for your kind and enthusiastic words. Now I would like to invite our mentor, guide and brilliant source of illumination, Dr. Archana ma'am, the President of Delhi Unit MSI. Please, ma'am, say something on this occasion.
Thank you. Thank you so much, Ankit. Sir, greetings to you first. You are our elder and our torchlight, torch bearer. And sir, I would like to just inform you that he is the younger version of Brahmajit Dahiya. He is called Ankit. Ankit Rohilla is his name. And actually, I also wanted to mention Brahmajit, which sir has done. Under able guidance of Brahmajit Dahiya, today Dahiya surname has a special meaning because Ravi Dahiya has bagged a silver medal for our nation. And so And actually all the Indians are very proud of it. So Dahiya rocks actually. So Brahmajit Dahiya is also one of those gems who has really worked very hard with his team. I would like to name Taranjeet, Mobina, Rakesh, Abhishek, and Satakshi, Charvi, all the office bearers, sir. And they are gradually transferring the legacy to their juniors who are conducting these things every time. Today also you will see 3 different people. Last time there were 3 other people, and next time again a team of 3 each time is coming forward, and all are, I would say, ek se badkar ek. So this is I wanted to tell you, sir. He is Ankit, and but we call it younger Brahmajit only because he has all the properties of Brahmajit. And all of these guys are very simple. They mean work. They are very honest, dedicated, well-mannered, and very knowledgeable. And these will actually surpass all of us, including today's speaker, Abhishek, Sachin, and all of us. This is our mission. We believe in them, sir. So as Ankit has already told about flow cytometry, without flow cytometry, we can't imagine immunology, any developments in immunology, cell biology, and the allied sciences. Mr. Pradeep is amongst us today, who is definitely our gem. We are very, very proud of him since beginning. He's all the way joined us from Perth, Australia online. After a proper office day work, today, sir, during midday, he rang me up on a video call and he showed me the flow cytometry that he was doing. I was so amazed and overwhelmed that I asked him to take the pictures and short videos and to include it in the session so that besides me, everybody can see. So it's an absolute homecoming for all my dear, dear alumni, and aap se hum hain. You are doing well and you are making us feel very worthwhile. We are very proud of you, and you know that I have always believed in all of you. So I wouldn't take much more time than this, and now I would request the convener, today's convener, Mr. Ankit, to carry forward the session. And thanks to you, sir, Deshmukh sir, for sparing your time. You coming and starting the session makes the session very lucky, useful, and so many people attend because they want to meet you here, sir. Thank you. And I'm really proud of my students, sir. Thank you.
Thank you so much, ma'am. Your words are always encouraging and motivating for us. Now to carry on the session forward, I would like to call Abhikash to give general instruction to the participants. Over to you, Abhikash.
Thank you, Ankit. Now, to ensure a smooth and hassle-free execution of our event, here are a few general instructions which are to be followed by all the participants. Firstly, I request you all to keep your audio and video turned off while the talk is going on. Secondly, if you have any questions or queries, keep on writing them in the chat box. They'll be surely taken at the end of the talk. Also, this talk is currently being live streamed on YouTube. So if you face any problem attending it here, then you may switch to YouTube and join via the link given in the chat box. With this, I wish all the participants an eventful and enriching session. Now I would like to call Anshita to give the official introduction of our today's esteemed speaker. Thank you.
Thank you, Abhikash. Thank you so much, Archana ma'am, for this opportunity. A very good evening to everyone present. I, Anshita, on behalf of Delhi Unit MSI, feel very elated to give the official introduction of our today's esteemed speaker and our super senior, Mr. Pradeep Kumar. Pradeep sir is a flow cytometry and equipment specialist presently working in Telethon Kids Institute, Netherlands, Perth, Western Australia. He pursued his BSc in microbiology from Swami Shraddhanand College, University of Delhi in 2009 and MSc in microbiology from Guru Jambeshwar University of Science and Technology, Hisar, Haryana in 2011. Sir has worked as a research trainee in AIMS, New Delhi on cloning, expression, and purification of human synuclein alpha protein, and in ICGB New Delhi on redox biology of Mycobacterium tuberculosis, flow cytometry, and BSL-3 facility. Pradeep sir has also served as flow cytometry operator in ICGB and AIMS New Delhi. Sir has also been field application specialist flow cytometry in BD Biosciences Private Limited India, where he has provided analysis and sorting support on JET in airflow cytometers across India, and also supported as a faculty in BDJH FACS Training Academy, New Delhi on required basis. Moreover, Sir has been technical personnel in KU Leuven, Belgium's largest and one of the most renowned universities in Europe. There, Sir has been involved in teaching, training, and troubleshooting for more than 150 regular users on 13 instruments for different manufacturers with experimental design, execution of flow cytometry, and also mass cytometry experiment. Currently, Sir is independently managing flow cytometry core facility in Telethon Kids Institute and providing a high-quality flow cytometry and cell sorting service. Furthermore, he is the member of Institute Biosafety Committee and also an active member of Institute Central Laboratory Management Team. We are extremely excited and we look forward to have an enlightening session with you, sir. The stage is all yours. Thank you so much.
Thank you everyone. Good evening to everyone. And I'd like to thank Professor Deshmukh to set up this different units across India it's a really good platform to present and to basically communicate and be in communication with everyone. So I thank everyone for organizing this, and thank you, Dr. Archana ma'am, for reaching out to me to give this presentation. It's been a while that we were trying to schedule it, but because of COVID and because of other things, like it's been rescheduled a few times, but finally we are here. And thank you, Ankit, thank you, Abhikash, and thank you, Anshita, for organizing this, and all the participants who are here for taking out time to join this presentation. So I'm Pradeep, and I look after the flow facility, and everything was introduced, like Anshita basically explained everything what I've did so far. So past almost 9-10 years, I'm just doing flow cytometry, and probably I'll be doing flow cytometry with some other things for the rest of my life. So I'll start with my presentation. Normally, this presentation for the basics, applications, and other things, it takes about 3 to 4 hours. So I had to struggle to bring these slides down to about like 40 to 50. But because of the time constraint, we can schedule it some point of time in future to have a full-fledged presentation if that's required. So I'm happy to do that at some point of time. So I'll share my presentation. I will start my presentation, and at the end we'll address all the questions. So in between, I'll play some videos which I was working. I was doing some experiment today. So, and I was talking to Dr. Archana, and then thought like at least we can make video what I'm doing. So it's not a crisp video, but at least that will give you how the instrument looks like, how the experiment is being done. So those sort of things. Okay. So I'll start my presentation now. Just a sec. So my presentation is visible, right?
Not yet.
Sir, you have paused from Google Meet. You go to Google Meet, resume your PowerPoint presentation. There is a button that written or click to resume the presentation.
Okay, just a second.
Yes, sir.
Yeah.
It is visible.
It is here.
It is here now. Okay. All right. So I'll start with flow cytometry, and I'm going to include basics, applications, and the employment opportunities that you can, like how to make a career in flow cytometry. And the same rules are basically apply for all other technologies as well. Okay. So plan of the talk is basically to include what are basics of flow cytometry, how we define flow cytometry, what information we can get from a flow cytometer about a cell or anything that we are running through instrument, and how does a flow cytometer work. So that will cover the basics of flow. The second thing will be applications, what are research and clinical applications that most of the time this technology is being used. And the third one, I'll be covering the employment opportunity, like what are the requirements of positions, what educational requirement you need, what sort of experience you need to make career in flow cytometry. So those things I'll cover. So starting with basics of flow cytometry, if you need to define flow cytometry, what it is, it's a technology that simultaneously measures, means it is doing multiple things at the same time, and then analyze multiple physical characteristics of single particles. So this single particle here is important. The reason is because this technology provides you information of single cells. That's why it's really important, because if you are interested in looking at multiple particles at the same time, then other techniques are also there, like microscopy. But because of this technology that it is able to do analyze single particles at the same time and thousands of single particles at the same time, that's how it's become important. And most of the time we run cells in flow cytometry as they flow in a fluid stream through beam of light, usually a laser. So as the name suggests, flow cytometry, flow means something is flowing, cyto means most of the time related to cells, and metry means we need to have the measurement of those what we are running through the flow cytometer. Okay. Generally, there are two types of flow cytometers. One are called flow cytometer analyzers, and the other one are called flow cytometer sorters. Okay, so the difference between these two instruments are analyzers basically analyze your cells and they give you some information. You get the statistics and then you analyze your data. And then everything, all the sample that you're running through the flow cytometer everything is going into waste. While the sorters basically analyze your populations the way analyzers are doing, but at the same time you can physically isolate some 4 to 6 small subpopulations into separate containers like Eppendorf tubes or your PCR tubes or 96 well plate, different platforms. So with sorters, you have physical isolation of cell types. With analyzer, it's just the analysis. So sorters does both things. And I'm going to show through pictures, like just two picture of two instruments. One is the analyzer, other one is sorter, which I have in my facility.
Sir, excuse me.
Yeah, please.
Sorry for interrupting. Can you please go for slideshow?
Full screen. Full screen.
Yes.
Yeah, better.
Yeah, perfectly.
So
thank you.
Most of the time you'll hear that people call that we are doing FACS, and it's a general term that people basically refer to. So fluorescent activated cell sorting. Okay, so it's a general term that we normally use for some kind of technology that we are doing, but actually FACS is a trademark of one of the manufacturers of the flow cytometers, which is BD Biosciences. But in general, normally people who are doing flow cytometry experiment, they sometimes call it like we are doing FACS. We are going for doing FACS. Okay. So the picture that you are seeing at the moment is basically of a sorter. So it means that it's doing analysis as well, and it's doing sorting as well. Okay. So from top, the main component of the instrument that you're seeing, that your cells are flowing through the instrument, but they're not flowing just by themselves. They are flowing through a fluid stream, which is called the fluid basically that running through the instrument the moment you turn the instrument on, which is called sheath fluid. So the meaning of the sheath in itself is to give covering around something. So the reason that why we need to use some buffer through the instrument is that because the light source that we use in flow cytometry are lasers, and they are really high power. So if your cells are directly exposed to laser, the lasers can definitely harm the cells. So we need to give some kind of covering around the cells, and that covering is basically of the fluid. So fluid is continuously flowing through the instrument, and then we basically ask through software, we command to the instrument to aspirate your sample through the center of that sheath fluid. So from the left side of the slide that you can see, there is a laser. So laser is basically aligned at one point, and then through from laser, one by one cells are flowing through the instrument. So the moment laser and cell interrogate with each other, it means the cell is hit by the laser, some light scattering happens. So flow cytometry basically employs the advantage of light scattering. So whatever light is scattered is basically collected by different detectors. As you can see, the first detector is called forward scatter. The second detector is like side scatter. So, and then there are the other different detectors which basically collect the light. So all these light basically are photons of different wavelength, and based on that you get different information. So I'm going to cover in further slide what sort of those information we get from flow cytometer, like information of size, information of granularity, and the information of fluorescence. Okay, so that we will cover in further slides. But so the moment interrogation happens, scattering happens, and then we collect that light. So all the analyzers instrument basically stop their work at that point, and everything then basically is going into waste. So all the cells, all the fluid is everything going into a waste tank. Okay, but if we are doing sorting, then what happens? These cells are basically flowing through a continuous stream. So what we do, we vibrate the stream in kilohertz by giving frequency. So the stream will break into droplets, and then individual drop may carry your cell of interest, may not carry your cell of interest. And we also give charge to stream as well. So when individual droplets form, they may carry your cell, they may not carry your cell, and they are charged. When they're passed through high voltage deflection plates, which carry about like 4,000 to 5,000 volts. So based on charge, positive and negative, they are sorted into individual tubes, and at the center, everything is going to waste, and few population that we want from mixture of populations that we can sort into either left or right, and then maybe far left, far right. And this way you can sort 3 to 4 population on the left and 3 to 4 population on the right. So that's the maximum that you can do at the moment. So this is as a whole what flow cytometry is, but we are going to cover these individual details, like what sort of information, how to transfer sample from your tube to the instrument, how you get the information, how the signals are generated, how the signals are collected, and how electronics of the instrument process your signal. Okay. So these are some of the contributions that achievement that led to flow cytometry development, because on itself flow cytometry couldn't be able to do everything by itself. So there are a lot of discoveries like this microscopy, the light scattering, and then staining, and then droplet deflection sorter. So all these different technologies, they were brought together to form an instrument called flow cytometer. So there were a lot of, the backbone of the flow cytometry basically comes from all these discoveries, and then they were put together to have a technology called flow cytometer and the instruments respectively. Now, I'm trying to compare it with microscopy, because it's not possible for everyone to see a flow cytometer, or during undergraduation it's really hard. But we do have access to light microscopes where we prepare the slides and we see cells. So how we can compare with flow cytometry with microscopy is that in a light microscope you need some source of light to basically see your cell. That light can be direct sunlight, that light could be a bulb, that light could be anything. So we don't need specific light. So comparatively in flow cytometry, the light source is always a laser. Now, why we need laser? Why cannot we use direct sunlight? Because we are talking about individual cells. We are talking about single cells. It means we need monochromatic source of light. We need the light of a single wavelength, which can basically hit individual cells one by one. Okay, so that's why we need specific lasers. Even in microscopy, there are some instruments like confocal microscopes where you need to use lasers as well. Now, you need some kind of collection lens in a light microscope to basically reflect light and then to basically collect the light. So same thing happens in flow cytometry as well. We need some kind of collection lens. We basically collect the light, because all the light that we are collecting is basically scattered light. So some collection lens, some deflection lens, and some optics of the instrument basically helps to collect that light. Okay, so the majority of the components are comparable, but at the same time they are different. But it's easy to basically compare different components of a microscope with a flow cytometer. Now, why we need flow cytometry if we can do so much with microscopy? One of the most important reason, one of the most applicable reason is basically that when we are looking for single particles, we are looking for single cell information. So it means we need to provide instrument single cells. We cannot run tissues, we cannot run body fluids directly. But in microscopy, you can put anything under the lens of a microscope and you can see your cells. Okay, so that's why we cannot. The prime requirement of the flow cytometry is basically you need to prepare first single cell suspension. If you are if you're taking a tissue, like if you have a cancer tissue, you need to first homogenize it, make single cell suspension out of it, and then you can run it in the flow cytometer. The other requirement of the flow cytometry is basically there is a size constraint. Like you can run maximum up to 70 micron of cells, like 1 to 70 micron is most of the range that you can run. So anything beyond that would be hard, and there are some specific instruments that are required to do flow cytometry on larger cells. Okay, but most of the like human blood cells, whole blood, they can easily be run on a flow cytometer. So I'm going to cover a few terms that are really important, like to understand the flow cytometry. So one of them is what sort of things you need. Can you directly see everything on flow cytometer by running cells? No, you just get few information if you run your cells directly, just the information of size and just the information of complexity. But if you are looking for fluorescence, then you need to provide fluorescence means like the fluorescence doesn't come from the cells, it comes from a fluorophore. Okay, so there are different type of antibodies. Now what these antibodies are, the difference between these and antigen is like our target is basically to look for the antigens or proteins. They may be present on surface of cell, they may be present inside of the cell. Now those antigens and proteins, we basically provides monoclonal antibodies for specific type of cells, and antibodies are going to bind the antigens, and then that antibody is basically conjugated to a fluorophore. That fluorophore basically gives you information about fluorescence. Okay. Now I'll take an example of something so that it's clearly understood. We'll take an example of, let's say, a person is basically having HIV. He's HIV positive. So that's a patient. So what happens in HIV? Basically, there is a reduction of CD4 T helper cells. So one of the immune subpopulation, which is CD4 cells. So those are T helper cells, they get depleted. And because of that, the patient is immunocompromised. So what happens for that immunocompromised patient, any other disease, they are really prone and they can deteriorate the conditions even further. So how flow cytometry is used in that case is like we can monitor health of those patients by monitoring the level of CD4 T cells. How? So if CD4 T cells are present, monoclonal antibodies basically are those type of antibodies which can specifically bind to only on the epitopes of the cells which express CD4 cells, if it is a CD4 antibody. Okay. Now that antibody, let's say it is conjugated to FITC labeled fluorochrome. So when there is a cell, on top of cell, there is, if the CD4 cell is, CD4 marker is present, then antibody is going to bind, and then that complex antigen-antibody complex will go in front of the laser. Okay, so the moment interrogation happens between cell and laser, the fluorescence that you get is basically coming from a fluorophore rather than from a cell. And if there is no marker for it, antibody is not going to bind, and during the protocol of preparation of your cells, it will be washed away. So on right side of the picture that you can see, there are 4 quadrants. So 1, 2, 3, and 4. So the lower left basically is negative for A, negative for B. It means there is no FITC labeled antibody, there is no PE labeled antibody. So FITC and PE, these are some fluorochromes like phycoerythrin, fluorescein isothiocyanate. So these are fluorochrome compounds. The number 3 is basically positive for A. It means only antibody A is present, only marker A is present and marker B is not present. So if you look at a bivariate plot, there are 4 compartments in that. So lower left is basically 1, which is negative for both. Lower right is 3, which is basically positive for whatever we put on x-axis. And number 2 is basically positive for 2, that's 2, positive for only whatever we put on y-axis, and number 4 is basically positive for both. It means if we see populations on a 4th quadrant, it means both markers are present. Okay. Now, what are the requirements of and information we get from a flow cytometer? Flow cytometer basically gives us 3 information of size, information of of internal complexity, and the third information is fluorescence intensity. Okay. Now the information of size is basically called forward scatter. So there are some specific terms that one need to remember if they are doing flow cytometry, and that which is basically covered during the training of the when the user is getting training. So why we call it forward scatter, why we call it side scatter. We'll come to that part in future slides. The third one is fluorescence intensity. So whatever this the fluorochrome compounds that you are seeing, like APC, L-phycocyanin, PE, phycoerythrin, all these are fluorochrome compounds that are employed in conjugation with antibodies to look for specific markers on cells. Now, the requirements of basic requirement of flow cytometry is if you are able to prepare single cell suspension from anything, whether it is tissue samples, body fluids, or anything like from marine samples, anything that you can use. So if you are able to prepare single cell suspension out of it, you can run it in flow cytometer, and if it is within the size of 1 to 70 micron. But there are some small particle detector option as well, which you can go up to 0.2 micron. So an example of this is like nanoparticle studies by flow cytometry. In the next few slides, I'm going to cover few the components of the flow cytometer. So all flow cytometer, doesn't matter if they are sorters or if they are analyzers, they basically have three components: fluidics, second optics, and third one is electronics. So the purpose of the fluidics is basically to reach out your cells from your sample tube. So you can, if you have your cells in sample tube, so from your sample tube, the cells, the fluidic component of the instrument reach out your cells to the point of interrogation. Point of interrogation means where laser and cell interact with each other. So the moment interrogation happens, part of the fluidics is over. Okay. The second thing is optics. Optics we divide into two parts. First, to generate the signal, and the second, to collect the signals. So to generate the signals, we need what we have in the flow cytometers are a light source, but these are lasers. And there could be only one laser. There could be up to maximum for now is like 10 lasers in a single instrument. Okay. So all these lasers are sitting somewhere in the instrument. So with the help of prisms, mirrors, we route these laser light to a common point where all these lasers are sitting on top of each other, like 1, 2, 3, 4, 5, 6. And then your cells are flowing one by one through each laser. Okay. So the moment interrogation happens, light scattering happens. Now, part of the excitation optics, which is to generate the signal, is over, and the second comes to collect the signals. Okay, so when I'm talking about signal, it means the photons of different wavelength. Okay, maybe from 300 wavelength or maybe up to 800, 900 wavelength, anywhere between that. Okay, so those are light signals, like photons of different wavelength. Now, the second thing is to collect the signal. Signal means all the light that is scattered by the cell, maybe through the surface of cell, maybe from the inside of the cell. So that scattered light is basically collected by a collection lens, like all the light is focused to a common point, and then through there, it goes to second component of the optics, which is called collection optics, where we have the detectors which can collect different light. Now, all the signals are going through particular cables, and the same cables like fiber optic cable, which are used to transfer photons of different wavelength from one point to another point as a medium of communication. So they may be carrying one type of wavelength, they may be carrying different type of wavelength. So how to segregate different wavelength signal, different kind of optical filters are used, like green filter, red filter in microscopy, or filters of particular wavelength which can transmit only particular wavelength of light. So once signal goes to those components, and then it is different signals are scattered, like you can see the blue, the green, the yellow, and the red. So all signals are segregated into different wavelength, and then they're sent to individual detectors, which are photomultiplier tubes in case of flow cytometers, and then they are sent to electronics of the instrument. So fluidics is to reach out to your cells to the point of interrogation. Optics, to generate the signal, we need lasers. We need some prisms, mirrors to route the laser light to a common point. And then we need some optical filters to segregate different wavelength of light, and we need detectors. So optical filters and detectors are a component of collection optics. Okay, so fluidics, excitation optics, correction optics, and then the third component is basically electronics, which convert these optical signal to digital signal. And when you see data of flow cytometry, you see data in the form of a dot on a dot plot, or you see in the form of a histogram peak. Okay, so that's how you. So that's a broad range of like what are different components. I'm going to discuss these components into detail in further slides. So the first component is fluidics. So on the lower left side, the tube that you are seeing is a kind of tube that you prepare your samples, like, and this on top of that tube, there is a filter cap. So it means you need to filter your samples before you run into the flow cytometer. So those are the tubes that are used in the flow cytometry most of the instruments. So you load your sample onto the sample port, and in video I'll show you how these samples are loaded. So on top of the top diagram is basically, if you look at the sample tube, the sample is being aspirated, and then there is a flow cell. So flow cell is the heart of flow cytometer. Flow cell is the place where all the lasers are aligned, and then your cell will pass one by one through each laser. So flow cell is the heart of flow cytometer. Interrogation happens within the flow cell, and then after analysis is done, everything is going into waste. Now, there are two types of fluid running through the instrument. One is called sheath fluid. So there is a sheath regulator for it, and there is which is in a tank. So sheath fluid is in a tank, and then air is given to the tank, so it is getting pressurized, and then sheath fluid from the tank will move to the instrument. So before the sheath goes to the instrument, it passes through a sheath filter, and then the same way sheath is managed, at the same way sample is also managed. So your sheath fluid is continuously flowing through the flow cell. On the right side, if you see, this is a cross-section image of a flow cell. So the outer, from the both sides, sheath fluid is flowing, and through the center of it, sample is flowing. Okay. So, and these both fluids flow parallelly without mixing with each other. So they don't mix with each other because they are maintained by hydrodynamic focusing. So in that case, what it means, like, then two fluids can flow parallelly without mixing with each other. Okay. Now, the bottom two image on the right side is basically one of a low differential pressure. The second one is a high differential pressure. Means you don't change the pressure of sheath fluid in the instrument, but you can push more cells through the instrument. So low differential pressure means 12 microliter of your sample per minute is flowing through instrument, and you can see one by one cells are flowing through. But what happens when you increase the flow rate, like you push more cells through instrument, like 60 microliter per minute of sample is flowing, then more cells are pushed, so cells start flowing in a zigzag fashion, and you may lose some resolution of the instrument, resolution of data. Okay. This slide is an example of the same thing, like low differential pressure and high differential pressure. So what these images are of a cell cycle peak. Like we know there are different stages of cell cycle: G0, G1 phase, S phase, G2, M phase, and you can do that cell cycle analysis through flow cytometry as well. So this is a perfect example of low sample pressure versus high sample pressure. You can see like G0, G1 phase in first picture where it is running at low sample pressure is 68.7%, S phase is 19.16, and the G2M phase is 9.56. But what happens when you push more cells through instrument? You basically, there is a compromise in the resolution of the instrument. And if you look at the G2M percentage in the second picture, is basically increased to 15.14. So that's not true. So it depends what kind of application you are doing with flow cytometry, and your different sample rate basically change according to that. Okay. The second component is basically optics. So I'm going to cover what I've covered in the initial slides, like there are two components of the optics. First one is excitation optics, which includes lasers and filters and mirror that route the laser light to the fluidic stream. The second component of the optics is collection optics, which includes fiber optic cables, because the way lasers are sitting somewhere in the instrument, because each laser has a particular size. Some are bigger in size, some are smaller in size, and there is power assembly attached with that. So they're sitting somewhere in the instrument. At the same time, detectors are also sitting somewhere in the instrument. So we need to transport the signal from the flow cell where the interrogation happens to the point of detectors. So we use fiber optic cables for that. So fiber optic cables can basically transmit signal from one point to the other point without losing the intensity of signal. Okay, so those are part of collection optics. Now there are different type of lasers can be used. That can be diode laser, air cooled laser, water cooled lasers. It means there are different medium how the lasers are prepared, but there are different like coherent sapphire, which is a solid state laser, and they have a different wavelength. So all these lasers are different wavelength. So you cannot change lasers from day to day. When you purchase the instrument, the lasers, the laser line of the instruments are fixed. Like this is an example, like there may be 4 different lasers, 488 nanometer blue laser and then the second laser is 405 nanometer. Now why these specific lasers are used, I will be covering that as well. Okay. Now, I talked about the optical filters, like to segregate different wavelength of light. If you look at the right three image, these are example of different type of filters that are used to segregate different wavelength of light. Like there is one fiber optic cable which is carrying signal of 500, which is carrying it also carrying signal of 550, it carrying signal of 600. So all these signals need to be segregated, because if all the signal goes to one detector, they simply convert all the signal and you will only get one data. But if you segregate different wavelength of light before signal goes to detectors, you can have different signals. Okay, so these are different type of filters. The first image you can see, it says LP500. It means it is a long pass 500. More than 500 wavelength of light will pass through it, and less than 500 wavelength it will block. The second one is, the bottom one is SP500. It means less than 500 wavelength of light it will transmit, and more than 500 it will block. And there is a third filter called BP500/50. So it is a band pass filter which only transmit particular wavelength of light. Means you divide this 50, the second digit, into half around first one. So 500 divided by 25, 500 plus 25, 500 minus 25. So 475 to 525 is the light it will transmit. Less than 475, more than 525, it will basically block. Okay, so these are different type of filters that are used to basically transmit signal from one point to another point, or before they are reaching to the detectors. That's the layout of that's how it looks into the instrument. If you look from the top, so there's a fiber optic cable that says like 2-. So the cable is basically bringing all the different signals of different lasers. The outer one that you are seeing as a black, which says like 700 nanometer, 610 nanometer, these are PMT detectors. And before signal is going to these detectors, you can see there are different barriers. So, and they are used to basically So yeah, I was trying to have the pointer. So if you can see, all the signals are from the fiber optic cable are bringing to the optical block. This is the whole optical block, and the signal, all these outer ring is basically of the detectors, which are photomultiplier tubes. Inner to that ring, these are basically optical filters. So you can see light from here basically covering different wavelength. From a 488 nanometer blue laser is first going to a filter, and then it is basically collecting light of 780 nanometer. Then the light it passes, but the light which is not of that filter is basically transmitted to a different filter, and that's how it is transmitted from one point to another point. And then with the help of these, all the signals from higher wavelength to the lower wavelength are collected and segregated and sent to these detectors, which process these photons of light to electrons. Okay. The third component is electronics of the instrument. So after the signal basically from photomultiplier tubes, they are sent to electronics of the instrument, so photon in, and then these raw signal are converted to digital signals by analog to digital converters. And with the help of digital signal processors, they are basically converted, processed, and the signals are converted. At the end, you basically see dot for each cell in the form of a dot plot or as a peak in the histogram. Okay, so each dot in a flow cytometer on a dot plot basically represents one cell or the kind of population that you are running. So the summary of components is basically three components: fluidics, optics, electronics. Laser light, your cells are flowing through the instrument one by one. It's interrogated by the lasers, and based on different laser like violet laser, red laser, blue laser, these different type of signals will be there, like these basically carrying signal of blue laser, that's from the green, that's for the yellow, and that's for the red, and that's for the violet. So all these signals are basically sent to the electronics of the instrument to give you data in the form of a histogram peak or in the form of a dot plot. So forward scatter, side scatter. So then the next thing we are going to cover is like what type of information we get, like information of size, which we call forward scatter, information of complexity, which we call side scatter, and the information of fluorescence, like information from FITC, information from PE fluorochrom. Okay, so this is an example of how like laser is coming from this path, your cells are flowing from top one by one, and the interrogation happens. Okay, so the properties of FSC and SSC, forward scatter, why we call it forward scatter? is basically because the light which is scattered in the forward direction is collected, and that gives an information of size. Now, how that information, how we get the information of forward scatter is basically, if a smaller cell is flowing through the laser, then from the surface of cell, light will be deflected in the forward direction. Okay, so it is that diffracted light from the surface of cell in the forward direction of the light. From that's why we call it forward scatter. And if it is a bigger cell, like bigger cell going through the flow cytometer through the laser, then bigger cone of light will form in the forward direction. You get bigger signal peak comparative to smaller cells. So based on the information or the light that you're getting from the surface of cell is a diffracted light, we call it forward scatter because it's happening in the forward direction of the light. Now, the side scatter is the reflected light. So it is coming from inside of the cell. So there are different type of cells. Some cells have more granules, like if you take an example of human whole blood, like eosinophils, neutrophils, they are multi-lobed. They have more granules inside them, while lymphocytes or your monocytes, basophils, they have comparatively less granules. So more granules, more light will be reflected in all the directions. Less granules, less light will be reflected in all the directions. But what we do not collect light in all the directions, which is coming from inside of the cell. We collect light 90 degree to forward scatter, and that's at the back. So that's why we call it side scatter. So forward scatter information gives us information of size because it is coming from the surface area from the surface of your cell. While side scatter information comes from inside of the cell, and that basically because of the granularity, less granules, less light, more granules, more light, and more light is collected. Because we are collecting it at 90 degree of the 90 degree to forward scatter, that's why we call it side scatter. Okay, so that's the first two information that we get: forward scatter and side scatter. So this is the same image, like smaller cell will go through, medium cell will go through, large cell will go through, and you get accordingly smaller, medium, and the big peak of the signal. Second information is side scatter signal, which is basically this cell has less granules, so less light will be reflected, while this cell has more granules, more light will be reflected in all the directions, but we collected light 90 degree to forward scatter, and that's how it is collected. So forward scatter signal gives information of size. Side scatter signal gives information of internal complexity or granularity of the cells. The third information is fluorescence, like how we get fluorescence and why we are doing this flow cytometry. I mean, what's the main purpose of it? Okay, so main purpose is basically fluorescence. Okay, because that we are able to detect fluorescence out of it. Now, how we get the fluorescence is any fluorochrome molecule. This is an example of a fluorescent isothiocyanate molecule, which basically absorbs light at a particular wavelength and which emit light at a different wavelength. Okay, so what happens when this FITC molecule, fluorescent molecule, is at the ground state? So what it does, it basically absorbs light at a particular wavelength. So when a wavelength of around 490 nanometer hits the fluorescent molecule, which is at the ground state, it moves from ground state to excited state. It means if we have a laser in an instrument which is close to 490 nanometer, and if this fluorochrome molecule, fluorescent molecule, goes in front of that laser, so that molecule will absorb light and then it will move from ground state to excited state. But it cannot stay for more than 1 to 10 or 10 nanosecond in that excited state. It has to come back to the ground state. So when it is coming back to the ground state, it releases some energy and in the form of heat or dissipated wavelength. Okay, so that's how you get the fluorescence. Now, how the loss of energy is relevant to wavelength is basically because energy and wavelength are inversely proportional. So when energy is low, wavelength is high. E energy is equal to hv, velocity, which is c by lambda. So energy and wavelength are always inversely proportional. So you can see when it absorbs the energy, wavelength was low, but when it emits the light, wavelength is higher. So we need to have a laser in the instrument which can basically excite the fluorochrome, and then we have some kind of detectors which can collect that light. Okay, so that's how you get fluorescence. Now, how we employ this technology in flow cytometry is because what we are interested in, let's say there is a cell and it expresses some marker like CD markers, cluster of differentiations. Okay, so those markers are specific, like there are about like 350 CD markers are already defined. So and some of those markers are specific for particular cell, like they will be expressed, those CD markers will be expressed on particular cells and not all the cells. Okay, so what we basically need, those CD markers are basically are antigens or proteins, and they may be expressed on surface of cell, they may be expressed intracellular in the cytoplasm. Okay, so these CD markers, those antigens and proteins, if we have an antibody which is specific for that particular antigen, if it is there, then it's going to bind that antigen. So antigen antibody interaction will happen, and then this antibody basically is conjugated to a fluorochrome. Now these antibody and the fluorochrome conjugation are available in market. Like there are different companies which basically sell the reagents for these experiments, and you can have different combinations of antibody with a different conjugation. Okay, now you can raise your antibody in your lab as well. Like you expose let's say your mouse to a foreign antigen, so the immune system of the mouse basically will develop antibodies against it, and then you can basically extract the organ from the mouse, and then you can purify that antibody, and then you can do that. But that's a very lengthy procedure, so most of the time people don't raise those antibodies, and they use commercial available antibodies tagged to fluorochromes. Okay, so these fluorochromes basically have two properties. One, they absorb at a particular wavelength, and the second, they emit at a different wavelength. So what happens what you can do with flow cytometry, there are instruments where you can use 40 to 50 different type of antibodies in a single tube. It means you just take one sample and you are looking at 40-50 different populations. 40-50 different population means there are different markers you are looking at. So different antibodies, different markers. And all these antibodies are conjugated to different fluorochromes. So more parameters you are looking at, more complex the instrument you need. Means more lasers you need, more optical filters you need, more detectors you need. Okay, so and that's the emission spectra of different fluorochromes. So if you read about fluorochromes a bit, like what properties they have, so you'll see like they absorb at a particular wavelength and they emit at a different wavelength. This is an example of different fluorochrome, most commonly used fluorochrome emission spectra. So if you look at the 488 nanometer wavelength, so all these fluorochrome molecules like FITC, phycoerythrin, and phycoerythrin conjugated with Cy7. So all these are fluorochrome molecules. They absorb same wavelength, but they emit different wavelength. Like FITC molecule emit near about 520, 530, while PE emit near about 570, 580. PE Cy7 emit near about 750, 80. So all these fluorochrome molecules are absorbing same wavelength, but they are giving different emission wavelength. Same way, this is like red laser, and there are different fluorochrome that absorb that light and emit different wavelength of light. You can see in all lasers, all the emission wavelength are basically higher than absorption wavelength, because emission wavelength is always higher because absorption wavelength is low. And when energy is low, wavelength will be higher. And then the third example is violet laser. So the instrument that I have in my facility, basically there are 3 instruments at the moment, and we'll get few more. But each instrument at the moment is basically having 5 lasers. So we have 488 nanometer laser, which can do 2 parameters. It means we have 2 optical filters for that laser. The red laser can do 3 parameters, violet laser can do 6 parameters. So that's how instrument configurations are designed. Okay. Now, how you get your signal? I mean, how these histogram peaks are basically forming. So that's happening because the moment cell enters the laser, some photons are absorbed and some photons are emitted, and you see some voltage peak. The moment the cell is at the center of the laser, maximum absorption, maximum emission, and you see maximum signal. So when it leaves the laser, absorption is less, emission is less, and that's how you see the voltage peak. Okay, so and we can quantify these signals. Basically, the maximum signal is basically height signal, and sum of all the height signal is basically area, and the time it is spending in front of the laser is basically width or time of flight. So that's height, that's whole thing is area, and that's the width at that time it is spending in front of the laser. So this is an example of a dot plot. That's a picture, like the first picture is basically a forward scatter light, means information of size. Going from left to right is increasing size, and side scatter on y-axis going from bottom to top is increasing complexity. So it means this cell is basically smaller than this cell, and this cell is basically having more granularity because it's sitting on top than these populations. So that's how you design it, you look for your populations. So this is a picture of basically human whole blood, where by experience I know these are lymphocytes, these are monocytes, these are neutrophils, because neutrophils have more granules and they are almost 10 to 12 micrometer in size. So they are sitting on higher than lymphocyte and monocytes, because the monocyte size is bigger than neutrophils, but at the same time the granularity is, the complexity is less. So that's how they are basically placed on forward scatter versus side scatter, means size and complexity of cells. Okay, and the red blood cells and the platelets, they don't they are of really less size and with no granules, they will be sitting over here. So you can see there are different signals like negative population, positive only for green, positive only for red. But if that cell is basically expressing this marker as well and this marker as well, then it's a double positive. So this is double negative, this is single positive, and double positive. This is an example of the same picture where we are looking for two types of cells, like CD3 is a marker for T cells. CD19 is a marker for B cells. Okay, so we are looking at lymphocytes. So those are lymphocytes, and this whole blood suspension, we basically added antibody which is conjugated to FITC for T cells, antibody conjugated for B cells that is conjugated to PE. So we added that into there, and all the cells that are positive for within that lymphocyte population are basically stained. So 74% in this population is basically positive for T cells. 9% is positive for B cells, and 17% is basically negative for both. They are neither T cells, they are neither B cells. So in lymphocyte population, we basically mostly have three subsets like B cells, T cells, and NK cells, natural killer cells. So most probably these are NK cells, but we cannot claim unless we put marker of NK cells. Okay. So flow cytometry, you cannot just cannot just predict. You need to basically add antibodies properly and then stain and then have results, and then you can claim. Okay, so these are example of different markers like T cells. The CD markers are CD3, CD4, CD8. Okay, and there are different examples for these, like for stem cells, for granulocytes, for oligocytes, T helper cell, T regulatory cells. So these are different type of population. So when you look at the details of C markers, you would be able to find what in. So these CD markers are specific for specific populations. So the next thing, so that's the basics of flow cytometry, which includes fluid components of the instrument, fluidics, optics, electronics, and the three information that we get from a flow cytometer, which are information of size, information of complexity, and the information of fluorescence. So all these all these information are relative information. Flow cytometry does not tell you like it is 10 micron, it is 20 micron. It basically tells you like this population is smaller than that population. This population have more granules. So it's all these information are relative information. It doesn't tell you absolute information. Okay. So, and it is a pure quantitative technique. It doesn't tell you what happens to a patient. Okay, like in cancer diagnosis, it doesn't. It gives you information. It gives you statistics, and then based on the phenotyping or based on the knowledge and the literature available, you basically tell that's a kind of disease it's progressing on. Now most of the flow cytometry is used extensively. And in any immunology lab or cell biology lab or more, flow cytometry is the most heavily used technology in any research lab, and it has a lot of clinical applications as well. So I'm going to cover some of the applications. So the research applications, clinical applications, stem cell applications, it does have application in plant biology. Initially, basically, flow cytometry started with plant biology. So before the concept of monoclonal antibody, before 1975, flow cytometry was primarily used just for plant biology, for doing cell cycle analysis, for calculating nuclear content of the cells. And it's also used in veterinary science as well. Okay, so there are three types of major categories that applications can be divided into. First is analysis, the one we called immunophenotyping, and nucleic acid dye applications fluorescent proteins applications, which like GFP, green fluorescent protein, yellow fluorescent protein. The other application could be like cell counting, like in the case of HIV patients monitoring, absolute counts are done where we know like the GPs, the general practitioner, they are giving treatment to the HIV patient, and then they're monitoring their health at regular point of time and seeing if the count of CD4 cells is going up or if it is stable or if it is going down. Okay, and the third one is the cell sorting, where you basically analyze your population and sort your subpopulations. Like with sorting, with analysis you can just see this, but with sorting, what you can do, you can sort this population into separate tube, this subpopulation into separate tube, this population into separate tube, and then you can do further experiments with those. So immunophenotyping is the most common application that normally routinely is done. So what is immunophenotyping is the process of identifying presence of antigens or proteins using antibodies. So whatever we were talking about, like antigen is there, antibody is there, fluorochrome conjugated is provided. So it is basically called immunophenotyping. So you look for the phenotyping of your cells, like you look for different kind of antigens using antibodies, and that's called immunophenotyping. And flow cytometry is not the only tool for doing that. There are other tools as well, but because this gives information on single cell level and gives you absolute information in terms of statistics, like in terms of percentage, then it becomes really important. There are different type of nucleic acid dye applications as well, like DNA ploidy. If it is a diploid, tetraploid, you can do that. You can do cell cycle analysis, like there's a cancer cell. So a normal cell having a normal cell cycle peak, while a cancer cell, it has a different cell cycle peak. Now you have given some anti-cancer compound to that cell, and after giving that, you are looking at what's that anti-cancer compound basically. At what stage of this cell cycle the cell cycle is getting arrested. So if the compound is effective, and if it is blocking the cell cycle of the cancer cell at, let's say, G1 phase, so you won't see anything beyond G1. So that's how you compare between normal and a cancer cell, and you can look for the cell cycle analysis in that. You can also detect apoptosis, which is the programmed cell death, like what cells are live by using flow cytometry, what cells are just went into early apoptotic stage, what cells are about to die, called late apoptotic stage, and what cells are into necrosis, or the cells are completely dead. So you can do apoptosis study with that as well. And there's some specific application called site population analysis as well, which you can use using different dyes. So you can use dye as well. All the nuclear stains, like a healthy cell membrane will not be compromised, and the cell is not going to go, the dye, membrane permeable dye, like propydium iodide, it's not going to go inside of the cell. So if it is not going inside, you will not see signal in the laser, and that's how you can say like the cell is viable. But if the membrane is compromised, then the dye is going to go inside of the cell, it's going to stain the nucleus, and then if the signal will be positive, mean the cells are dead and the remaining cells are live. Clinical applications of flow cytometry includes like absolute CD4 counts in terms of HIV or AIDS patients, and into arthritis as well. Like you can diagnose the arthritis, joint pain through flow cytometry. It's called HLA-B27 assay. A major application which is used in clinical settings is basically blood cancer diagnosis. All the leukemia, lymphoma, if it is acute leukemia, it is a chronic leukemia, what stage of the leukemia is. So there are different marker panels are basically used to diagnose this. So in diagnostic labs, there are flow cytometers which perform these, they basically get samples. So what a physician, pathologist basically do, by looking at the blood counts, they're suspecting that this patient can maybe having a cancer, a type of blood cancer. So they basically take some blood sample, and then they process with different markers. And if in cancer, we know one type of population basically is more than, and we know what percentage of normal population in a normal individual of those cells are. And when we run those cancer, the sample or the blood from a cancer patient, few type of populations will be more, other type of populations will be less. And based on that, you can say because all these cancers are already defined, like acute leukemia basically will be having these specific markers, other ones will be having those. So based on that statistics, percentage positive and negative, more or less, then the pathologists basically decide, okay, that sort of treatment we are going to go ahead with this patient. And then you can look for solid tumors as well, like you can basically extract solid tumor from the patient body, and then you homogenize those tissues and prepare single cell suspension out of it, then look for some markers, stain, and then that's how you look for the solid tumors. And there are primary immunodeficiency disorders applications are also there. You can look for hemolytic anemia, bleeding disorders, and recently, I mean, from quite some time, for about like 5 to 10 years, these are used in cross-match assay, like where the transplantation is going of a particular organ. So donor and recipient cross-match is being done, that they are compatible to receive or transfer the transplant or not. So those studies is called like flow cytometry cross-match. Flow cytometry in plant biology is majorly used for DNA content analysis, ploidy level, and then cell cycle analysis. And you can also determine the nuclear genome size by using different dyes. You can look for different options, like you can look for a cell cycle from a plant leaf. Basically, you use a lysis buffer, you chop the plant leaf into smaller sections, and then you filter those sections and you get cells from it. And then you basically stain it with a dye and look for how the cell cycle or the nuclear content or the genome content looks like. In veterinary science, it's basically mostly used for sperm sorting, like the pre-selection of the sex in the cattle of higher milk yield, like X sperm sorting, Y sperm sorting, and different kind of basic and medicinal research, like immunophenotyping for all the veterinary studies. Some other applications includes like stem cell and nanobiotechnology, marine biology. So flow cytometry is basically employed in kind of everywhere. So it's just like, as long as you are able to prepare single cell suspension from anything, like from marine samples. I've seen one instrument, they are already taking it in flow cytometer in the sea, and they basically take water sample from different parts of the sea and then characterizing different populations, like to check the microflora of the populations in there. The next is I'm going to include some of the pictures and video from the facility which I was working today, and then I thought like maybe I could show you some. So this is one of the analyzers. So that's how it looks. This image is basically of the sample tube that you load your sample tube in there. This is basically here in there, and this tank is basically of the fluid tank called sheath fluid, and those are the commercial tanks that we basically purchase, and then we refill this tank. So the moment we turn the instrument on, sheath fluid will start continuously flowing through, and then from this tank it is going to the instrument. And the second tank is basically of the waste tank, so everything that we are running through the instrument, it's coming into this. So that's the waste. The third image basically is of the sorter. So we keep this instrument within the hood, because all the sorting experiments, we cannot afford contamination of whatever cells we are running through. So we basically keep the instrument within the hood, and that's called one of the sorters. And there are different sorters with different companies as well. Okay, so that's the image. So what you can see here, I opened this part, this thing. And once you open this, that's how it looks like. Okay, so in analyzers, you load your sample from bottom, it is going onto top, and then everything is going into waste. In sorters, your sample goes from here, the stage is loaded up, and then from this capillary, it is going into this flow cell. So that's how the flow cell looks like. So the lasers are coming from this end, all the lasers aligned on top of each other, they are coming to the flow cell. And then your sample is coming from the top, and one by one cells are flowing through. And then after the analysis is done, we basically use some cables to basically charge the stream. These are the cables like piezoelectric device. So we charge the stream, we vibrate the stream. So continuous stream will basically break into droplets. And this is the image. Like until this point, the stream was continuous, but because we have vibrated this stream so much that it starts forming drops, individual drops, and now these individual drops may carry your cell of interest, may not carry your cell of interest. The white thing between a drop is not a cell, it's just the image. Okay, so don't confuse yourself with that's a cell. So these individual drops then goes in front of these high voltage deflection plates, basically, and then based on the charge, positive and negative, they are sorted onto the left or onto the right, and this is the center stream where the continuous stream is going into waste. The next slide will be is a image how continuous stream is basically going into droplets. Okay, so and you can see there are 4 appendopes in there. So what I was doing today, I was running a patient's whole blood sample, and in that sample we were looking for different populations. Like there was a population of basophil panel and dendritic cells. So there were a lot of populations we were looking at, like T cells, B cells, and basophils and monocytes, those sort of populations. And then of all those different populations, we were interested in just 4 populations, and we wanted to segregate them into individual tubes. Okay, so that's the inside of the flow cytometer. That's how it looks like. Okay, so this is the sorting block, and this is the stream will go from here, and then there is a camera in here, which basically takes the image, and that's the image of the stream that you see. Now, these individual drop carrying your cell or not will go through these deflection plates and sorted based on electrostatic attraction and repulsion to the left, to the right, to the far left, to the far right. Okay. Yep. So that's the video. Like, I need to play it again. So when I click a command on load, so the software takes a command to load the sample tube from the instrument to the point where it starts aspirating your sample. So I clicked on load, and then I took the image like its stage is going up, and then basically start aspirating your sample. In my presentation, I'm going to include different components of the flow cytometer. So those components includes fluidics, the second component is optics, and the third component is electronics of the instrument. So the main purpose of the fluidics is to basically reach out your cells from the point of interrogation. I mean, when the cells and laser interact with each other. So that's happening in the flow cell assembly. So flow cell assembly is basically point where your cells and laser interact with each other. So the moment interrogation happens, your part of the fluidics is over. So the fluidic component of one of a flow cytometer I'm going to show you now is basically, that's the fluidic component. So that includes the buffer of the system that instrument keeps running over. So everything that we are running through instrument is basically coming into waste, and that's to maintain the temperature of the cells and the collection cells into the system. And then there are different tanks, cleaning tanks that are used to basically clean the instrument afterwards. So we don't want anything to leave biological material inside the instrument. So we need to clean up after every experiment. Okay? So the moment interrogation happens, your fluidics, your optics is divided into 2 parts. So the first is to generate the signal. Generate the signal means the scattering of light, and that includes lasers. So that's the source of light that we use in the flow cytometers. So, and then those signals are basically sent to the second component of the optics, which is called collection optics, where you collect the signals. Signals means photons of different wavelength that are coming from the fluorophore. Okay, so these fluorophore absorb wavelength, and then they emit wavelength. These fluorophore are basically conjugated to the antibodies. Those antibodies are monoclonal antibodies. It means they're going to bind to the specific cells, and they're not going to bind if the cells, they're not going to bind if the marker is not present on the cell or inside the cell. Okay. So like CD3 marker, CD4 marker, CD8 marker, these are markers of T cells, T helper cells, T cytotoxic cells. So those are different type of populations that you look at. So those are different 4 streams. So when we basically put the voltage to the deflection plate, so you can see 4 streams are going into 4 individually appendop tubes, and the central one is the waste. So the cells which we don't want will go just to waste, and the deflected population will go into the individual tubes to sort. Here you can see different populations based on the size and based on the complexity of cells, and we basically get these cells, apply this population to the next plot, and then we exclude the doublet, the cells which stick together, we call them doublets on the basis of size, and then we apply this population to the next plot where we see doublets on the, exclude doublets on the basis of complexity and granularity of cells. Next to it, we are looking for populations like for CD3 marker, which is tagged to a fluorochrome called Alexa Fluor 700. So it's a fluorophore that absorbs some wavelength of light and emits some wavelength of light. So CD3 is a marker of T cells, CD19 is a marker of D cells, and it is tagged to fluorochrome fluorescent isothiocyanate. So based on these two markers, we in this plot we are looking at 4 different populations. The population which is this population, which is negative for T cells as well, and it's negative for B cells as well. So here is the population which is positive only for x-axis population, which is T cells. So these are T cells, and the population here is just positive for B cells, which is CD19 positive B cells, and the population which is here would be double positive. So we don't expect any double positive populations in a mature blood, because all the lineage for all the cell populations are already divided and then they are moving forward. So based on that, then we are doing further gating and we end up sorting basically 4 populations. One is this, 2 are these populations, and one of these populations and one of these populations. So our target is to basically sort 4 populations, and that's the statistics that you get, how much percentage population is there, those populations are positive. Okay, so I'm going to give command acquire. It will start splitting again, and you will see number of cells per second are flowing through instrument. And then I put the voltage on the deflection plate. Just hold on a moment. Sorry. So then I'm going to give instrument command to basically start sorting the cells. So this is the basically continuous stream that's going into waste. But then if I give the instrument command sort, and if I click on OK, then it basically starts sorting different populations. And you can see basically there are different, based on which population is more positive or more available in your sample and which population is less positive. So accordingly, populations will be sorted. So the center stream is basically an illuminated stream by a laser light, and then the side populations are basically the population that we are sorting. So one is this population, the second one is this population, the third one which you'll see just once in a while because that population is very rare. So you don't see much of those populations. And then the 4th population here. So at the moment we are sorting 4 populations. That's P7, which is this population, and then P10, which is this population, P9, which is this population, and then P8, which is this population. Okay. So we are sorting for these 4 populations, and we are sorting these into appendobes. So you can sort into different type of devices, like in 96 well plate, 384 well plate, in a flask, in different tubes, in appendobes. So at the moment we are sorting 4 population, and in this we maximum want 400,000 of cells. So once it reaches to 400,000, it will stop on its own. Okay. So that's the time it is taking. That's the flow rate of the sample that it is flowing. So at flow rate of 1, it normally aspirates 18 to 20 microliter of your sample that you are running. Now the image that you are seeing over here is the image of the stream. So your cells are flowing through a continuous stream. The stream is basically of a buffer that's running through the instrument the moment you turn the instrument on. Okay. So, and your cells are basically traveling through the center of that fluid stream. So after analysis means the moment your cells and laser interrogate, like the cells are hit by the laser, interrogation happens and some light scattering happens. So that light scattering is basically the signal that you are looking for. Okay, so that light scattering is collected based on the detectors in the system, which are part of the optics. So forward scatter signal, side scatter signal, fluorescent signals, all those are collected, and then this continuous stream with the help of frequency in kilohertz, like 42.2, so it means 42,200 drops are forming per second. Now maybe not all drops carry your cell of interest, maybe they are carrying your cell of interest. Okay. So, and we basically, we vibrate the stream. So at some point, the continuous stream will break into droplets. So not those droplets may carry. So the sample that I'm running is about. So the sample that I'm running is about to finish because I can see it through here. The tube is about empty. So I'm going to stop the sorting, and the moment I stop sorting is basically stop the acquisition. And when I click on unlock tube, it's basically you'll see the tube will come down and then the sample is unloaded. So I'm going to take that sample and then I'm going to put this into this tube. Right, so that was the experiment that I was doing today. So I just I tried to capture it. It's not a very crisp video, but I thought like at least it will help a little bit to understand what sort of experiments. I just realized that I took too much of my time. So I'm going to quickly.
But it was, Pradeep, it was wonderful. It helped us to understand the practical aspect. We really appreciate that you did it spontaneously today and you are making it available to everybody from a distance. Thanks a lot. Now you can move ahead. Yeah.
So I'm covering last component, which is the career opportunities. So end users of this technique are academic and research institutes like medical research institute, all sort of academic institutions, and hospitals or clinical testing laboratories. So where clinical testing is being done, so where cancer testing is being done. So you'll probably find those laboratories, and those labs are basically opportunities as well, where you can basically have a training and then basically learn a little bit of flow cytometry. And the third one is basically pharmaceutical and the biotechnology companies that basically employ. So all sort of research labs where they're doing some sort of cell biology, plant biology, or flow cytometry is being involved. And it is just not the flow cytometry, like there are other techniques as well, which you can make your career out of. Okay, so like microscopy, like in genomics, like single cell sequencing, and there are a lot more like mass spectrometry. So there are a lot of techniques. They are limited like 5 to 10 techniques that you can basically focus 1 or 2 of them, and then you can successfully make your career out of it. So flow cytometry facilities basically comprises of instruments, and they can be really expensive as like $1 to $1.5 million as well. And to run these facilities basically and support research, expert personnel are required, which basically have expertise in one of the technologies, and we basically run and manages those facilities. Like it's not possible for the researchers to do basically everything. So they know a bit part of it. Like in my facility, the sorting is basically done by me, because at most of the places sorting is these instruments are a bit complex than normal analyzers, and basically staff are running those. And I provide training to users, so I train them, and then they can basically use by themselves. So why these technology experts are important to the field? Because these facilities are big. Sometimes some facilities are like 15 instrument, 20 instrument, 30 instrument. And to maintain those instruments, to train the users, to train new users, to do experiments, to design these new experiments, experts are required. So and those people who are specifically doing just this are basically managing these facilities. Now, the applicants with direct exposure to these techniques are few. So as a part of PhD, users do a lot of these techniques, but sometimes they do one of the techniques more and they make career out of it. Sometimes they do less, but it's not possible for everyone to basically learn everything. And most of the researchers who are doing PhD or after PhD, they look for postdoctoral scientist position or different research opportunities rather than technical positions. But you can basically make career at any stage of your career, like even after bachelor's, after master's, after PhD, but the opportunities are comparatively less just after bachelor's, but most of the opportunities basically need at least master's. Why I included this slide, like what are the companies which are making the flow cytometers or associated reagent of the like antibodies or other things, is reason that just for flow cytometry, there are about like 18 to 20 companies which are making flow cytometry. So it means there is so much of scope. And this is just for flow cytometry. There are other instruments as well. Some of these companies are making, or there are some other companies which are making specific microscopy instrument or single cell sequencer or different technologies. So there are a lot of options available with each technology. So it doesn't feel like just after your education that would you be able to find right position or not, or how to find positions, but it is there. So some of the companies are basically have major chunk of business from all over the world, like for flow cytometry, BD Biosciences and Beckman Coulter basically share approx 60% of total market share. Okay. Now, what kind of low profile you basically get in core facilities, core labs, or industry, or in medical research institutes? Like in flow cytometry core facilities, most of the hierarchy of positions are basically you start with a junior technician or a operator, or you are a research assistant and you learn bit part of flow cytometry or other technique, and then you look for position specific in core facilities. And then it comes to senior technician or flow cytometry specialist or facility manager or facility director. So these title of these positions are basically are with respect to different institute, because each institute having different different way of segregating these different titles of positions, but it mostly starts with junior technician, senior specialist, or facility manager or facility specialist kind of thing. In companies, we basically can work as a technical service provider, as a field application specialist, as a field application scientist, or senior application. So that's how I was in BD Biosciences when I was field application specialist. So if I would have stayed there, I would have been to basically this point or this point at this stage. But eligible for same roles in sales and marketing as well. Like I know there are people like who after the age of 40, they were masters in science and they were doing the application part, but then they decided to do a diploma course in management, and then they went into marketing or sales. So there are different roles with your experience that you can move into. Okay. In pharmaceutical companies, most of the time it's like core facility manager, scientist, research scientist positions. So flow cytometry specialist or scientist. So these sort of positions are available everywhere. Then what are the requirements of these positions basically? So it varies completely, but most of the positions, like most of the companies, will require at least post-graduation. If you are choosing after bachelor's, there are options, but there are very limited options. You probably would be spending same time, like bachelor's plus 7 to 10 years of experience, or master's plus 4 to 6 years of experience, or a PhD with 0 to 3 years or 2 to 4 years of experience. If you are choosing after your master's or post graduation, you can look for initially working as an operator. That's what I did. Like, and sometimes it takes just a year to move to companies on a higher role. Sometimes it takes 2 years, 3 years. So it took me about 3 years, but I know there are different people who basically were there in India for just after their master, just for a year in a core facility. And then they moved to basically, they moved to like 2-3 people I know, they moved to Europe on junior technician position, and just within a year on that position, now they are having like 4 to 5 different senior positions somewhere. So it takes about as a first-hand experience of me, like it takes about 2 to 3 years to make any career out of it. But if you are joining after PhD, then you need direct experience of extensive flow cytometry or any other technique, and then during your PhD, and then the vendors from different companies, they basically know you were doing sort of experiments of that technique, and then they can basically invite you, or you can apply through them, or you can join different companies or core facilities. Now, where to get the training and courses in India? Okay, so the first link is basically International Society of Advancement of Cytometry. So that's that's a universal site, like where all the workshops, all the international conferences that happens, you can find details in there. TCS is basically the Cytometry Society of India, where you can look for different workshops or conferences, and majority of it, like if you're a master's student, you can still look for it. Sometimes they give undergrad students as well, like you can join some workshops or conferences. NCBS basically have a core facility, and they have a C camp where they continuously provide different courses on flow cytometry and microscopy as well, because I know someone who basically is doing a lot of training. And there's a ATPC Advanced Technology Platform Center that is in the regional center of biotechnology, that is in Faridabad. So it's in NCR region. So they have a whole lot of, probably they basically have all of technologies, and you can approach, and there are different ways to basically get training. If you know someone your senior is already working there. That's how I learned. I basically started my flow cytometry in ICGB, and I started learning one instrument. I basically learned all the instruments, sometimes with the help of my colleagues. Some of my seniors helped me a lot as well. So that do happens. You can look for training in different things as well, like AIMS, electron microscopy department. Each year they conduct 1 or 2 training sessions specifically for scanning, electron microscopy, or TAM. Like there are different options in there as well. And this is just for flow, but there are same other techniques as well that where you can have the training, or a 6-month course or a 3-month course. They are available. Okay. And BD Biosciences, they have an academy in Jamia Hamdard University. We call it BDJH FACS Academy. They do regular course for the flow cytometry, and I think they're still doing 6-month flow cytometry course as well. I'm not sure. I mean, it was there for quite a few years back, but they still are doing regular flow cytometry basic course, which it's a 3-day course which you can join. So there's a payable fee in there, but I think it's worth it if you wanted to make career in that particular field. Now the job sites, it's very common. People do ask me like where we can find jobs for it. So there's a site called flowcytometryjobs.com, Seek, LinkedIn, Indeed. These are common websites. There is a Purdue University flow cytometry laboratories. They also have a job page where the jobs are listed, and you can basically type anything like flow cytometer operator jobs, flow cytometer specialist jobs, flow cytometer scientist jobs. Sometimes they need really experienced people who basically have PhD and they have a lot of research experience, and they want to employ their expertise into research. So they have like flow cytometry scientist position. Okay. So, and there are different cytometry society as well, country-wise, and you look for job options in different pages. Talk to your colleagues and seniors. You'll definitely find them everywhere, probably in every field, because I mean, even if it is just the Microbiology Society in Swami Sadanand College, there's still a lot of seniors that are basically everywhere, and they did help me at some point. I mean, they definitely had me at some point, so it was really helpful. Now, I basically looked after some positions. The idea to basically include some screenshot is basically to look for what are the minimum requirement for the position. Like this is a position like scientist flow cytometry, and I mean, there are thousands of positions as well, but if just focus on what requirement they have, like PhD with minimum 0 to 2 year experience of flow cytometry, or a master's with 4-year experience, directly relevant experience, or a bachelor's with 8-year experience, so preferably 10 years of that. So you can see, like if you're applying just after bachelor's, it takes more time. If you're applying master's, after master's, it takes less time. If you're doing after PhD, it takes even less time. So it's up to you at what stage you want to make your career out of it. And then there was another position, basically scientist or manager flow cytometry in a pharmaceutical company. And then you can say PhD in immunology related field with 2+ years of experience, or a BS or MS with 10+ years of experience. So at some point, your education, like your PhD, can be managed with experience, but it takes almost same time or comparatively more time as well. But I mean, there's no specific rule for it. Some positions need less time, some positions need more time. Another example of flow cytometry technologists, which basically just need BSc degree in biological sciences, but all these positions outside India, it looks like they have bachelor degree with some experience, but in our case, the best is basically to have at least master's, because you can get job in India with a bachelor degree, but there will be a limit because you are being paid based on your qualifications. Like they cannot underpay you, they cannot overpay you outside India. Okay. Then there was a flow cytometry co-director position where you need like be just bachelor's with +7 years of experience. So you can see like it's still a co-director position, but they just need bachelor's in a related field and plus this much experience. But if someone is PhD with 2-year experience, they will have preference. But there are so many positions, and some instruments are specific, like they say experience with BD influx. So it's a jet in air flow cytometer. If you have experience with that instrument, your application will be preferred over others. And flow cytometry specialists, like you need at least 4 years of experience with bachelor's. So different roles, different positions based on level of position, junior level, mid-level, senior level, manager level, director level. Based on that, your experience is required. Yep. So those are some of the examples of the job positions. before going to acknowledgement section, I would like to mention that it takes time. It takes 2 to 3 years at least to make career out of it. If you are really active, really aggressively applying everywhere, that's what I did. Like I got my first Belgium position in 2018, but I basically started applying in 2016. So it took me, even with 5-year experience, almost 2 years to find positions, because for few positions I was rejected after interview. So, but then I learned from it, and then at some point you'll get multiple positions offered. Okay. So if you have any question, you can talk to me, you can reach out to me, email me, and then, or you reach out to your seniors, even in India or outside, and they'll definitely help you. So moving forward, I would like to acknowledge all the organizations that I've worked with: Telethon Kids Institute, which I'm currently working, BD Biosciences that I've worked, and my master's from Guru Jambesho University, Catholic University Leuven in Belgium, and yes, of course, the Swami Shadanand College, which I basically started my everything from there. I would also like to thank Dr. Amit Singh. So at the moment, he is in Indian Institute of Science. So he was in ICGEB. So he did help me a lot, because I basically upfront like I wanted to do this flow cytometry, and he supported me a lot with that. But I mean, so my work was to basically do the lab work for flow cytometry, and whatever time I spare, he basically allowed me to learn and or to teach. So I basically reported him, and he helped me a lot with flow cytometry, because flow cytometry for myself, we basically started with Dr. Amit Singh Lab. So I want to thank him. And yes, it cannot be possible without your friends, without your juniors, without your seniors. So some of the pictures. So that's my colleagues, Sachin and Kuldeep. So he gave presentation last week. This was most of our batch during graduation, and I think it's the farewell pic from graduation. Looked very different. That's Abhishek, Kuldeep, and then that's me, the Yogesh, Sachin. And yep, Sadhna, Kriti. So those are our seniors from Shadanand as well. And I'd like to thank everyone, because yeah, we are still in contact, everyday contact, and it really helps a lot to take burden of everything. And yeah, friendship is always knowledge. Yeah. And without most, yeah, Dr. Archana, she, I mean, there would be, the words are definitely less saying about her, because she always motivated. It was just because of her at that point when we were in college that we felt like we were a bit, that we can get back our confidence, or we can do something, because it was really hard. The challenge when we came to college was not was to the knowledge of subject, but more was to speak. Like we couldn't speak at all. Like we cannot say anything. Like we were so shy, so less in confidence. But she really helped a lot. And then, yeah, by the end of 3 years, we felt like we can definitely do something. So thanks to you, man. It wouldn't have been possible without you. Yeah, you did help a lot, and I really appreciate. And you're still helping basically every time I reach out to you, and you're just there always. Yeah, nothing can be better than that. Thank you so much.
Thanks a lot.
Thank you, sir.
Thank you. And I probably took a lot of time. I think that is the biggest presentation so far. So I'm so sorry about that. But then it's really hard, ma'am, to include everything.
Thank you,
We
sir.
do understand, Pradeep. My sessions, even my routine classes also tend to be long, just because we want to give the maximum. That is the main motive. So that's okay. And it is a great evening spent with you. And even I am blessed to have students like you. Thank you. And I want to say a lot, but I will refrain so that others get a chance. So there is a question answer session. The questions will be moderated by Abhikash. Thank you. Thanks a lot. God bless you, Pradeep.
Thank you.
Thank you, ma'am. Thank you, sir. It was a truly wonderful presentation. The way you explained flow cytometry, starting from the ABCs, the basics of it, and you covered each and every component proficiently. You talked about the applications, the future prospects. So it was a complete and power-packed presentation. And the video that you showed us, that gave us a glimpse of your workspace. So that was the cherry on top, if you will. And the impact of your presentation is clearly evident, as the chat box is flooded with questions. So we'll see how many questions we can cover. So without any further ado, we may move to the Q&A session. So Are you presenting something, sir?
No, I'm not. I think I need to close the presentation probably.
Yeah, so this first question is by Shivangi. She asks, while the sample is passed from between the sheath stream, how come the sheath fluid and sample do not get mixed up?
Okay, so what happens? It is achieved by hydrodynamic focusing. Okay, so if you are able to maintain hydrodynamic focusing of your fluids or the cells that you are running, then two flow can flow parallelly without mixing with each other. So that is somewhat related to the Reynolds number, which is if it is less than 23 or if it is more than 23 at one stage. If it is well maintained, then the flow is well maintained, and then it doesn't flow, it doesn't mix with each other. And the design of the flow cell is like this. It's a conical flask. If you throw a long wooden block in a river, the moment the river bank comes, is basically the wooden block basically automatically aligns to the flow of water, and that's how it's flowing parallelly. So the outer covering is of the sheath fluid, the inner is basically of the sample. So it's basically achieved by maintaining the laminar flow. So if you are able to maintain the laminar flow by adjusting the pressure of the system, it can be achieved, and that's how it is achieved. They never mix with each other.
Okay, sir, thank you. Dr. Srivastav sir wants to say something. Sir, please go ahead.
Oh, hello Pradeep, how are you? After a long time. Yeah, I thought you were in Belgium, but came to know that you have moved to Perth. Very happy to see your success and other things, right? So the company that you're working on, what is the name? Telethon or something? So what?
So it's a medical.
It's a medical. Okay.
It's a medical research institute that is located into Perth Children's Hospital. So it's a full campus of different medical research institute, the university, and the hospitals. So yeah, all sort of diseases related to kids. The institute is working on, and then we have the research facilities here.
Very good. So Pradeep, I just wanted to say that, you should tell the students about it, that since you're working in a children's cancer hospital, a lot of hematopoietic stem cell transfers are being done for leukemia patients and other things. So flow cytometry plays a very, very important role in that. So you are sorting the cells out and trying to select the cells, the stem cells and things of those kinds. So I think you never touched upon that. So you can give that as an application here so that the students are made aware of it. And also what is happening these days is in cancer, people are trying to look for cancer stem cells. So we are trying to look for that also, and in that also flow cytometry plays a very big role. So I think you can address these two issues as well to the students, because I teach them in cell biology, so I thought maybe this would be pertinent. So if you have some time, maybe you can just... And also a little about the gating, because the gating concept, people are not very clear about exactly what gating is. So these are some of the terms that keep coming and students are a little confused.
Yeah, so I think the best would be that at some point I do a different session, which basically will be, it will be a long session, because a lot of applications like the gating, it takes time to understand.
Okay, yeah.
It basically comes from, like, if you are running a whole blood, you basically first to look based on the size and complexity that these are different type of cells, and then you first identify them based on size, and then you go from higher population to lower population. Like you first get from whole blood lymphocyte, then you get different type of cells like B cells, T cells, NK cells, and then you go sub-phenotyping of those B cells like and T cells like T regulatory cells. So these are different type of cells. So I think at some stage we probably need to schedule another session, because it takes a bit of time to explain the gating.
Yeah, I can understand. Okay, we'll try to arrange. You can request Dr. Archana to do that. Yeah, thank you.
Yes. And about the stem cell studies are being done in the hospitals as well, in the stem cell transplants as well, like where the stem cells are being used to transplant. So stem cells are sorted and then they're transplanted into the patient to basically have the cells that are moving from the early stage of their lineage and then overcome the cancer cells. So I can cover those applications as well later. Yeah.
Thank you, sir. So the next question is by Chitrali Roy. The question is, Is there antibody specificity for forward scatter and side scatter?
No. So for forward scatter and side scatter, you don't need any antibody because those information are just coming directly from your cell. So either from the surface or from the inside. That doesn't have to do anything with antibodies or the fluorochrome molecules. You can run just the sample without any fluorochrome molecules, and you can basically differentiate based on different type of cells. Just the size and information you can, granularity information you can get from it, and you don't need any antibody for those. It basically comes from blue laser 488 nanometer.
Yeah, thank you, sir. So Chitrali ma'am, we have taken one question from you. So for now, to give everybody a fair chance, I'll take question from others first. And if time permits, then we'll come back to your remaining questions. So the next question is by Paramjit Bhaiya. And the question is, sir, how flow cytometry can be helpful in studying different apoptotic stages?
So apoptosis is most common used, like one of the applications that can be done. So it just needs few antibodies. Like there's one dye called propydiamidite. So that stains the dead cells. There's another dye called annexin 5, which can be conjugated to a fluorescein molecule. It can be conjugated to a phycoerythrin molecule. So when you put these antibodies tagged to fluorochromes and you do the cells, the dead cells basically propydiamidite dye will go inside and it stain the nucleus, and you'll see them as positive. So in that case, positive signal means the cells are dead. So, and the annexin dye basically are the dyes that is used to differentiate between live cells and early apoptotic cells. So when we talk about the 4 quadrants, so the lower left will be live cells, and then one quadrant will be having early apoptotic, double positive will be late apoptotic, and then one will be for the dead cells. Within just apoptosis, there are different type of, you can look for the mitochondrial changes that happens in the cell. So within apoptosis, there are 5 to 7 applications in flow cytometry.
Okay, sir. Thank you very much. The next question is by Dr. Kriti Tyagi ma'am. The question is, can you mention a few examples where FACs have been used for identifying and quantifying the parasitized blood cells? Forward scatter.
Yeah, I need to look into that for the parasitized blood cells, because that's from Pradeep. I can reach out to her, but I need to look for the specific example that it's being.
Hi, Pradeep. That's
Hello.
Absolutely fine, and so nice to hear you. I want to quickly add something. Your presentation was wonderful. I mean, you touched everything precisely, concisely, and yet in a comprehensive manner. Very, very nice presentation. And our association has been very long. Ever since you joined the college, and I know I can always count on you, Pradeep and Sachin, and you have done so much for me specifically in comparison to what I have been able to do, and I know that I can always count on you people. So I wish you all the best, good luck, and wish you much success in your work.
Thank you.
It's so nice to see you here on this platform. Really wonderful feeling to see you here. And thank you to Archana ma'am for getting you here.
Thank you.
I am enjoying your reunion. I am
Nice to you, ma'am.
In fact, I got in the teachers group also, because Dr. Kriti is our colleague now. So I written there yesterday night that so तुम्हारी बहुत तारीफ हुई वहाँ पर. And Kriti also wrote that I'm proud to be a senior of this person. And Dr. Sumit was also here. I think he's still here. Abhishek, besides Abhishek, Sumit, your junior, and Kriti, your senior, everybody. Yeah, thank you. So nice. Bless you.
Thank you. Thank you, ma'am. Thank you, Pradeep.
Thank you, Kriti. Thank you.
Thank you, ma'am. Thank you, sir. So the next question is, by using flow cytometry, is it possible to distinguish cells that have been infected with lysogenic virus in which the viral genome is integrated in the genome of the cell?
Can you repeat that, please?
So the question is, by using flow cytometry, can we distinguish those cells that have been infected by lysogenic virus in which the viral genome is integrated?
Yeah, as long as we are as long as we are able, because those cells are integrated with virus. So what we can look after is if we are able to identify what sort of those cells are, then yes, we can, because in that case, we are not looking for identification of the viral in there, but if we are able to find what sort of those cells are, then when we can look for different antigen or proteins for them and that are present on the surface that the virus is in. So, and then we look for those protein and specific antibodies, and then we can stain those cells and can identify, okay, that's the cells, those are there from, and then we can sort those cells and do further analysis. It is possible, but it would be on the cellular level rather than on the virus level. So our target would be to basically look for the cells, proteins or markers rather than on the virus.
Okay, sir. Thank you very much. So the next question is by Richa Chandra. The question is, how do we decide which antibody to use using forward scatter to find any specific macrophage from the macrophages derived from bone marrow?
Okay, so bone marrow cells or the macrophages are monocyte derived. So what you can do, forward scatter, side scatter, you don't decide like which antibody or the fluorochrome you need to use. You basically look for the literature to find out what proteins are there antibodies available, and if you are able to have, if you are having antibodies, then you can do different type of stainings even if it is not available commercially. What you can do, you can look for indirect staining where you have one primary antibody and then you can tag it to secondary antibody and look for those markers. So there are specific panels available, I think, for macrophage derived populations that we can look into. Okay, sir, thank you. Sir, if your schedule permits, then maybe we can take a couple more questions. Sure, please. So the next question is, will there be any discrepancy if the sample is not freshly prepared? Yeah, so most of the, in clinical setting, most of the samples are recommended to prepare within 48 hours of their collection, and they should be well maintained for their temperature. So that's for the clinical one. For after staining is done, best recommendation is to run your samples within 4 hours of your staining, but it depends on the samples as well. Sometimes some samples are stable, like if you fix the cells and you stain them and you prepare them, and then you can leave them overnight at 4 degrees or the recommended temperature for your particular cells, and then you can run them. But most of the times user prepare, because those experiments are expensive, they try to do experiment and run their samples right after they prepared them. So the recommendation is basically to run as early as possible within 4 hours, or if the cells are not fragile, then you can, and if they are fixed, you can prepare, you can put them overnight in 4 degrees, and then you can next day. But if the human whole blood sample, 24 to 48 hours is time of staining that you have to stain, and after staining you can leave it for 24 hours if it is a clinical sample from a human blood. All right, sir, thank you. So now I think this is the last question for the day. So Pulkit asks, sir, what are the parameters considered for choosing fluorescent compounds for labeling antibodies? Okay, so your fluorochrome compounds are chosen based on the instrument configuration, like what your instrument is capable of. That's the first thing that you would consider before. So what happens, like first you identify what populations you are looking at, then you identify what antigens, proteins are there that you are looking at, then you know what markers are there. Now to choose fluorophore, there are different options available for each marker. Like for T cells, there will be like 50 fluorochromes available, but you are going to choose fluorochromes which you can run in your flow cytometer. If my flow cytometer can do only 4 fluorochromes, then I cannot choose anything else like apart from those 4, because molecules will not be absorbed and you will not get any signal. So you need to have the instrument configuration first in your hand, and then you need to decide how many populations you want to look, like how many subpopulations you are looking at. If you are looking at 15 different populations, you need to design your population panel in a way that it works really well, because not all fluorochromes are same. Some fluorochromes are really bright, some fluorochromes are moderately expressed, some fluorochromes are dim. So what you will do for all the bright fluorochromes, you will put your antibody with a rare marker. The population that is really rare, like just 0.1%, let's say positive. So I don't want to put that 0.1% antibody to a fluorochrome which is which is dim, like, because it's already dim, the separation will not be really good. So for the rare population, I'll put those markers with the bright ones, but I need to make sure that I have the configuration of the instrument first, and then you design your panel. Most of the time, if you're not really, if you don't have so much experience with flow cytometry, people from the technical specialist or application specialist from reagent companies, or someone in your team or someone in your institute, they basically help you. Like, I design a lot of panel for my users in the institute. So because they don't have enough experience with that, but it's a mutual conversation between researchers and the technical specialist, and then they come up with different options, like this we can do, this we can do, and then it's tried. And sometimes it works 100%, and sometimes it not. Sometimes we have to change the conjugation, like a marker with a different fluorochrome, because the more markers you are using in a single tube, more is the emission spectra of these fluorochromes spill into each other. And that's another thing that's called compensation. So that signal need to be clearly separated. So compensation is the most important part in flow cytometry. I didn't cover it today, but probably at some point, if that happens again, I'm going to cover it into detail, like what compensation is.
Thank you very much, sir. So we are done with all the questions, and I must say that we are looking forward to have another terrific session with you. So thank you very much for answering all the questions so patiently and gracefully. Thanks a lot. Now I would like to hand the stage to Ankit to give the official vote of thanks. Thank you very much.
Thank you so much, sir, for this comprehensive talk. I'm sure students have really benefited a lot. So before moving to official vote of thanks, I want to ask Dr. Archana ma'am, if she want to say something, please go ahead, ma'am.
Actually, a lot of time has passed. I can speak 1 hour about him and about such students. I will just let it suffice to say that he is a fine human being, very respectful student, and a blessing to be with. At the same time, a very much motivating senior and an accomplished speaker. Very accomplished, so fluent, so clear, so sincere. Somebody who can just record it in extempore and put it in a few hours to you after a working day. Now it must be 10 p.m. for him, but he is all smiles, all ready for everybody. So such people make the world go around. Pradeep, you and your friends, very proud of you. Keep it up. I have written a big comment there. You can see that. And thank you. Thank you. We'll meet soon again. Thank you. Thank you, Ankit. Very well convened. Abhikash, Anshita, very well done, children. Proud of you.
Thank you, ma'am. On behalf of MSI Delhi Unit, I would like to propose the official vote of thanks to our guest speaker, Mr. Pradeep Kumar, sir. Thank you, sir, for accepting our invitation and coming here today for enlightening us with your vast knowledge and experience in flow cytometry. I am very thankful to Professor Arvind Deshmukh, sir, and Dr. A. Archana ma'am for their encouragement and guidance, and also for giving us wonderful platform to gain knowledge. I am very thankful to Dr. Nitin Chauhan, sir, for his support in hosting and recording the session, and also Vishal Panchal for YouTube live streaming. I am thankful to all the student coordinators, our seniors, teachers from different colleges, and participants who have joined this webinar. Now we have come to an end of this session. Thank you once again to everyone. Stay safe, stay happy. Jai Hind. I would like to say to Vishal, please stop the live streaming and let's have a group photograph. Those who can come on the camera.